Thanks, Josh and everyone. I'm so excited to be here to kick off this exciting meeting today. Let's see if yes. Before I jump into the science, I want to thank the many funding sources that support my lab and the work that you're going to hear about today, in particular the NIH and the Blue Cone Monochromacy Families Foundation, along with the FM Kirby Foundation. And I want to acknowledge the many people in my lab who make this work possible. Everyone pictured here has contributed to the work today, and in particular, Ray Warner was responsible for a lot of the analysis that you'll see. And I'd also like to acknowledge my faculty collaborators, Tomas and Art. So I'm going to jump right in and start by talking about normal-sided humans being trichromats. And as the name suggests, this means that we have three types of cones that contribute to our color vision. These cones are aptly named the L, M, and S cones because of their long, medium, and short wavelength sensitivities. The L and M cones comprise 90 to 95% of the cones in the human retina, with the S cones making up only 5 to 10% of the total cones. In addition, the S cones are excluded from the foveal region, and their peak density occurs at approximately 1 degree eccentricity. So blue cone monochromacy, as this name suggests, occurs when patients have only one functional cone type contributing to their color vision. This is this is caused by mutations in the OPN1, LW, and MW gene array, with deletions either involving the locus control region of the gene array or a missense mutation in the CIS203 arch that affects both the L and M copies of the gene. And patients then present with reduced color vision and reduced acuity. They can have a nystagmus, which is uncontrolled eye motion, and many times they are myopic. So the goal of today's talk is to investigate the cellular phenotype in patients with blue cone monochromacy. To do this, we'll use a technique called adaptive optics imaging, where we first measure and then correct for the optical aberrations of the eye, and then that allows us to take a high-resolution image of the fundus. So shown here is an adaptive optics montage comprising just the small region outlined by the rectangle on the conventional fundus photograph. And with the adaptive optics, we can effectively zoom in and have high resolution that allows us to see individual cone photoreceptors in the living human eye. So the bottom left picture is the cone mosaic at the fovea of this normal-sided control, where each bright spot is an individual cone photoreceptor. And as you move away from the fovea out into the macula, you find the cones become larger, and also the rod mosaic is interspersed between the cones. And so on the image on the bottom right, the cones are the larger reflective regions, and the rods are the smaller cells. Adaptive optics is just a tool that we can use to increase the resolution of our imaging device. And so you can think of adaptive optics as a tool that allows us to do in vivo microscopy with really high resolution. Like microscopy, you can alter the detection schemes of your imaging devices in order to capture different modalities of images. And so, as described here by Drew Scholes in 2014, in addition to capturing the confocal light, we're also able to capture the non-confocal light and then split that non-confocal light into two detectors and look at the difference between those two collections. In doing this, we can actually capture two images simultaneously, one being the confocal image of the wave-guided photoreceptors, as shown on the left, and then the split detection image on the right. And that split detection image shows the cone inner segments. In a normal control, these cone inner segments then correspond one-to-one with the wave-guiding cones in the confocal image. So we're going to use these two imaging detection schemes now with the adaptive optics to investigate the photoreceptor phenotype in blue cone monochromats. We recruited 8 BCM patients to participate in this study, and 3 of those individuals had the deletions on the locus control region, while 5 had the CYS203 ARG mutation. 2 of those individuals had high eye movements that precluded montaging in the adaptive optics imaging. So the other 6 then were deemed successful for the adaptive optics imaging, meaning that we were able to create a montage over the central macula, and that yielded a 75% success rate for the adaptive optics imaging. I'm now going to show you the images from one patient. So this is the adaptive optics montage over the central retina and the macular region that we imaged. And we can now just zoom in to look at that structural phenotype of the cone mosaic. So again, shown on the left is the confocal image of the wave-guided reflectance, and the image on the right is the split detection image showing the inner segments. Salient feature in the confocal image here are the dark gaps in the wave-guiding mosaic. To make it easier to visualize, I've identified some of these gaps with red arrows. If you look between the red arrows of the confocal image on the left compared to the split detection image, you'll see that each of those dark gaps also corresponds with a cone inner segment. In addition, however, there are other inner segments visible in that split detection image that correspond with wave guiding cells. Those are demarcated by the blue arrows in this image. In addition, we can visualize the wave guiding rods in the blue cone monochromat, and those locations now are shown by the yellow arrows. All right, so what about the foveal region in this person? If we zoom in now and look at the foveal region, what you again see is a subset of cells in the wave guiding mosaic being highly reflective, showing that wave guiding property. But at the very center, there's limited to no wave guiding. And this does correspond with the central foveal area that does show cone inner segments on the split detection. And these images are representative of the qualitative structural phenotype of all 6 of the BCM patients that participated in the study. So with this central image now, we then wanted to quantify the phenotype that was found. And so every one of the wave guiding cones was selected, selected where the cone had both wave guiding properties and a visible inner segment. And then we used a sliding window to determine the location of peak density for the wave guiding cells, as located here with that red box. So I'm just going to zoom in now to that box and show you the confocal and split detection image at the top, just at that small region of interest. The image on the bottom left is now a pseudo-colored combined image where the confocal wave-guided signal is shown in the orange or yellow color, but the split detection image is shown in the blue. And this can just help visualize there that there really is a subset of these cells that exhibit the wave guiding. We already had identified the wave guiding cells, which are noted in the bottom right by the yellow dots, but then we went over this region of interest and identified all of the cones that were present as noted by the blue dots. And so with these measures then, we can calculate cone densities. So for this individual, there was wave guiding cell density of about 4,600 cones per millimeter squared, with a total density of around 29,000. We can do this now on all 6 of the patients that participated in the study and get cone densities for the wave guiding cells and also the total number at that location. And those numbers ranged from about 2,000 to 4,600 for the wave guiding densities versus 7,000 to about 30,000 for the total cone densities. And just to put this in perspective, the wave guiding densities for these individuals is about what you would expect for normal S-cone density. However, the total number of cones is reduced from normal. So how do we interpret this information? Well, there's a number of hypotheses that could be used for this information. This is work that is certainly still in progress. But right now, I think the leading interpretations of this data is that the waveguiding cells represent functioning S cones, and the non-waveguiding cells represent non-functioning remnant LNM cones. And certainly there's a lot of work yet to be done, in particular targeted functional testing to really tease out the details of these hypotheses and test them. But this is how we would interpret the data currently. Should those hypotheses turn out to be correct, there are implications then for treatment approaches such as gene therapy that would aim to restore function to cells that are structurally present. So with that, I will pause and thank you all so much for your attention.
All right on time, Jessica. Thank you. Any questions, please? Gus.
Jessica, really super. If you look at a patient serially, would you have an increase in the number of non-wave guided lines?
Yeah, sure.
Going out of segments first and then the increasing.
Yeah, great question. I think the question is, it really is blue cone monochromacy progressive? And would we see continued decrease in the photoreceptor densities and percents of waveguiding? That is still to be determined. All of this data was collected cross-sectionally. I guess, standard answer for BCM is that functionally these patients seem to be static, but if you look at the clinical imaging, it appears that there could be progression there. So we'll wait and see as these folks return.
Other questions? Yes, Elena.
So on the slide where you had listed out the total cone density for the different patients,
Mm-hmm.
do you know why there was like such a difference? Like one patient had 29,000, it looked like, where others had 8,000. Would that be difference in age, the mutation leading to their form of blue cone monochromacy?
Sure. Yeah, so fantastic. The question is, why is there such a range in cone density, and is that related to the patients? So we have a limited number of people contributing to this data, but I will speculate. It does appear that the patients with the deletion of the locus control region had a lower number of total cones. That would be something that could be tested with more patients, but that, I believe, is a decent hypothesis. And then age probably does have some, there's a hint of age there. Yeah.
So it's interesting that the cones that were destined to become red or green don't become blue. Do you think there's any way to potentially tweak the development so that those could become blue?
I mean, that's a really fascinating, interesting question. As you know, in development, the S-cone's fate is determined early, and so you would certainly have to intervene early. And maybe I'll leave my speculation there.
Therapeutically better to have the red and green cones restored if possible.
Absolutely, yeah.
We have time for one more question. Olga.
To continue on your question, this was a mutation in the remnant cones, right, in these cases.
Mm-hmm.
What if you knock them out? Then maybe the glucose will take over.
Maybe.
Is there
Wait.
anybody done that? I don't remember.
I don't know. I'll just say that. I don't know.
Okay.
We certainly can't knock out the LNM genes in our people subjects.
All right. Thank you very much, Jessica, for an excellent talk.
Thank you, guys.